channel, you might notice something a little bit different today because I'm not here with my co-creators. We decided that being a health and wellness channel and a channel that's dedicated to fitness and increasing our life expectancy and living life to the fullest, it wouldn't be socially responsible for us to film together right now because of the global pandemic situation. But hang in there, guys. Keep talking to your friends on social media because we're all in this together. We can get through this pandemic together. Today's video is a bit different. We're not doing a workout and we're not making kale salad or kombucha. What we're actually going to do is we're going to talk about telomeres. And I may hear you thinking, what the helomere is a telomere? But I can assure you this video is not clickbait. This video is science, and this video can help you improve your life expectancy, and it might even help us live forever. Telomeres were actually separated for the first time by a female Australian scientist, which is amazing, femme power. Her name is Elizabeth Blackburn, and I was fortunate enough to meet Elizabeth Blackburn in 2015. I went to the Elizabeth Blackburn School of Science, and to be honest, I think for that I should just get my degree now, or at least a pass in genetics. For her research, she actually won a Nobel Prize in medicine, and she was the first Australian female scientist to win a Nobel Prize, and that was in 2009.
Telomere DNA contains simple G-rich repetitive sequences arranged in tandem repeats. These sequences are specific to the action of the telomerase, with telomeric DNA requiring telomerase for complete replication. Telomerase is a specialized cellular ribonucleoprotein reverse transcriptase. This means that it can copy a short template sequence within the RNA component of an enzyme in order to add simple sequence repeats to the chromosome ends. Therefore, telomerase is synthesizing the telomeric DNA strand that runs 5' to 3' towards the distal end of the chromosome, extending.
The science of longevity genes is constantly developing, with it recently being discovered that there is a connection between telomere length and mortality in humans. Approximately 25% of variation that has been found in the human lifespan is determined by genetics. Some of the main genetic factors that affect the variation of the aging phenotype are involved with the basic maintenance and function of the cells of the body, in particular the maintenance of telomeres, DNA repair, and protection of cells from damage. Other genes that play an important role in longevity are associated with blood fat levels, the cardiovascular and immune systems that help to reduce the risk of common causes of death like heart disease and stroke. It was found in a study conducted in 1971 by Alexei Ovsyannikov that the connection between telomere length and mortality in humans was due to the incomplete replication at the end of linear DNA molecules. Additionally, this study found that women have longer telomeres than men, which explains why women have a longer lifespan. DNA polymerases are incompetent at being able to copy the 30 ends of linear DNA molecules. Therefore, eukaryotic cells need compensation for the terminal loss of DNA from the chromosome ends, e.g. the telomeres. These eukaryotic cells cap their DNA with many repetitions of telomeric sequence, short non-coding regions, in order to avoid losing critical genetic material. In early embryonic development, stem cells and germline cells contain high levels of telomerase, a specialized enzyme that provides telomere lengths. However, in somatic cells, telomerase levels are low and cannot prevent the gradual shortening of telomeres, resulting in a limited lifespan.
As mentioned earlier, certain gene variations in the cardiovascular system can affect longevity, and it has been found that there were shorter telomere lengths seen in patients that suffered from coronary heart disease and cardiovascular risks, such as obesity, compared to those that did not have this. Additionally, shorter telomeres were found in individuals with diabetes and psychological stress. Therefore, during these investigations, it was suggested that cellular aging can be indicated by telomere shortening.
More studies into science of telomeres have contemplated whether psychological functions like pessimistic orientation, stress, or depression are linked to the rate of cellular aging. In one particular study, it was found that shorter telomere length was linked with higher levels of pessimistic orientation. The effects of stress-related neuroendocrine activation can be amplified due to the pessimistic orientation, stimulating negative reactions to events that are stressful. Therefore, telomeric DNA damage in telomere sequence is provoked due to the neuroendocrine activation elevating oxidative stress. Consequently, the enzyme telomerase synthesis telomeric DNA, and therefore the damaged DNA may induce the reduction of surviving telomeric enzymatic activity that is needed in order to synthesize telomeric DNA. Hence, the telomere shortening is accelerated.
To summarize, it's actually these tiny little things on the end of our genes affected by reverse transcriptase nuclease, telomerase, that are going to affect our life expectancy and that are going to help us extend our life expectancy. And if we can manipulate these via science, we're going to be able to live forever. But I'm not a scientist, and I don't know if anyone watching this channel would be a scientist. I feel like they're way too smart for that. So there's other simple ways that we can increase our life expectancy through telomerase manipulation without having to get into a lab and pipette things and Bunsen burners science. And that is simply to reduce our stress levels, which is probably easier than it sounds, and during global pandemic is very hard. But if we can work on that, then you can decrease the rate at which your telomerase is being used up by the body and potentially increase how long you're going to live.